It's been great so far. I really like being outside. It's cool to continue learning about hydrology and the park itself.
I'm Cassidy Esposito, and welcome to Outside Science Inside Parks. We're here at Curecanti National Recreation Area. It is home to 3 recreational reservoirs and scenic river adventures. Today we will follow a water monitoring team as they work to collect water samples and document river algae. Let's take a look.
I'm Lucca. I work as a hydrology intern for Black Canyon of the Gunnison National Park and Curecanti National Recreation Area. We went on the boat to work with the USGS team as part of a 2-year partnership with them to collect samples all around the Blue Mesa Reservoir. So when we go out on the boat, we usually have 2 teams to do 2 different types of sampling. We have 2 people working the Van Dorn sampler, which is just a giant cylindrical tube that we lower into different depths of the reservoir to collect samples so we can see the algae and test for different things back in lab.
So we're getting a water quality sample that will be analyzed for metals, nutrients, and cations and anions. We're getting these samples at different depths to kind of learn how the various constituents are cycling and how they might be affecting the algae growth.
So in the park, there are these algal blooms that have been getting worse throughout the years. So we're doing sort of ongoing monitoring to make sure that we can figure out why they're getting worse and how to sort of implement change. Algal blooms can become toxic if the temperature gets too high. So when people swim, it can cause problems. They can get sick through E. coli or other cyanotoxins. The other team uses a YSI meter, which is just a meter that we lower into also every 1 meter.
His full name is Sir Yukon Inferno, YSI. We call him Yuki for short.
It has different probes that test for pH and temperature and dissolved oxygen. And so we check it every meter until we get to the bottom. And it gives us a representative sample of the whole reservoir.
SPC is 233.6. So we're taking data over a 2-year interval of the whole basin and getting info on why the harmful algal blooms are causing them to be toxic. So any data that we can get on temperature and pH and all of the data points that we get from this meter kind of give us insight into why the blooms are happening and what we can do to try to make them stop.
One thing that I was surprised about was that there's a lot of different types of sampling. So some days will be big boat days and we'll go out on the reservoir and use different kind of probes. But then some days we'll drive to a stream and hike in and wade the stream with our meters and get different types of data. One of the other projects we're doing is a cyano monitoring project. So we come out and take a grab sample with a little net that has a filter at the end of it, and then we put it through a microscope, like a cyanoscope, and we can put our phone on to take pictures of the slides. And then those pictures get sent into iNaturalist so we can identify the species of algae in the reservoir. In the lab, we do processing of the samples, so we filter them through our machine and add different additives like nitric acid and sulfuric acid so we can send them to the bigger USGS lab. It's definitely an all-around experience, and it's great to be in a team too, because a lot of field jobs I've been out in the field by myself. So it's nice to get to learn hands-on and get to see how they do it and then get to do it. It's like a lot of different projects all in one, which is sort of awesome.
Thank you for watching this episode of Outside Science Inside Parks. We'll see you next time.